Today we are very thrilled to welcome Dorothy Roberts, who's going to be discussing ethical challenges that arise when using race, ethnicity, ancestry, and other population descriptors in genomics research. My name's Holly Fernandez Lynch. I co-lead the Research Ethics and Policy series with Steve Jaffe. We're both housed in the Department of Medical Ethics and Health Policy, and you'll see in a moment that the series has several co-sponsors. Next month, if you can be enticed to join us again, we will be hosting Daphne Marchenko from Stanford University Center for Biomedical Ethics for another genomics talk, actually, so hopefully it will be of interest to many of you in the room. Daphne is going to be talking to us about the ethical implications of social and behavioral genomics, and you can see our full list of speakers for this series at our website. We also continue to add potential speakers to the list of people that we're hoping to invite in the future. So please share with us suggestions for people that we should think about inviting, especially individuals from diverse backgrounds or who can contribute diverse perspectives to issues relevant to research ethics and policy. Just a quick reminder about some resources that are available through our department, some online resources. So we have a series of short research ethics courses that are available for free continuing education credit, some of which can be used to satisfy your research ethics training requirements if you're doing IRB regulated research, both at Penn and at CHOP. I mentioned our many co-sponsors, so thank you to all of them, and thanks to Lisa Bailey, who made sure we got rid of the divider, found our speaker, right? So it was really critical to making sure that everything runs smoothly. And now I'm really pleased to introduce today's speaker. Dorothy Roberts is an acclaimed scholar of race, gender, and law. She joined Penn as its 14th Penn Integrates Knowledge Professor, with joint appointments in the departments of Africana Studies and Sociology, as well as the law school, where she holds the inaugural Raymond Pace and Sadie Tanner Moselle Alexander Chair. She's also the founding director of Penn's program on race, science, and society in the Center for Africana Studies. Professor Roberts' work focuses on urgent social justice issues in policing, family regulation, science, medicine, and bioethics. In 2017, she was honored by election to the National Academy of Medicine, and she served as a member of the National Academy's Consensus Study Committee that was charged in 2021 with reviewing and assessing existing methodologies, benefits, and challenges to using race, ethnicity, and ancestry, and other population descriptors in genomics research. So that report came out last year. That's the work that we're going to hear more from Professor Roberts about today. Just a quick word about how we'll handle questions and discussion. We will leave plenty of time at the end for that. We have folks joining us online as well. If you have questions and you're online, you can feel free to post those in the chat. We can read them out or call on you at the end of the session. We're also recording this session, so the video will be posted on our website as soon as it's available. And with that, I will turn things over to you.
Thanks, Holly, and thanks for all of you for gathering today. I'm really happy to be over at the medical school. For some reason, every time I come over here, it's like a maze, and I think I'm recognizing two people who I was behind as I walked. If I'd only followed you two, I would have gotten here, but I used GPS and it sent me to the back. I could see you all, but I couldn't come through the wall. Anyway, I was asked to talk about the National Academy's consensus report on the use of population descriptors, especially race, ethnicity, and ancestry in genetic and genomic research. So that's what I'm going to talk about. I'm going to first just say a little bit about my orientation to this question, because that's something I've been working on, writing about, talking to lots of medical students and genomic scientists about for over 20 years. And so just to give you a little idea of how I came to this project. I usually, when I talk about this topic, talk about the racial invention of science and the scientific invention of race, and want to make it very clear that race is an invented sociopolitical classification of human beings that came out of kind of ironically the Enlightenment period, the origins of modern Western science, borrowing concepts of race that were theological and that were invented in order to justify enslaving African people and conquering other indigenous people, dispossessing them, exterminating them, and the need for a theological explanation of why it was all right for Christian people to do this to other human beings. And that explanation was that God created human beings into different races and made white people superior to all other races, and Black people the most depraved and violent and uncivilized and all of that. And that was a really convenient justification for slavery. And when the Enlightenment period developed, the Enlightenment scientists, a vast majority of them, simply adopted a creationist view of the origins of human beings with the rankings and the idea that race is a natural division of human beings. And that idea has been central, absolutely foundational to Western science from the 1600s or late 1500s until today. The reason for it, the reason it's so powerful is for the same reason why Thomas Jefferson adopted that idea of race, because it explains away the power-based reasons for racial inequities, and especially for white domination and white supremacy. So Jefferson was able to say the reason why a nation founded on liberty and equality and tolerance and all these lofty principles didn't apply to Black people who could be enslaved, in fact, he enslaved his own children, was because of nature, not because of anything wrong with the idea of enslaving people, but it was just an unfortunate consequence of nature and the natural division of human beings into races. So when the human genome was mapped, there was a lot of, and I'm of course skipping over several centuries now of a contestation over this idea, but there was a relief that everyone declared, Francis Collins declared it, Bill Clinton declared, and Craig Venter declared, everyone at the top declared it disproves that race is not a natural division of human beings. There's no such thing as race at the genetic level. But no sooner had that happened than I began to read front page stories on in the New York Times about how the next mission after the Human Genome Project would be to look for genetic differences between races. And if you followed Nicholas Wade during this period, even before he wrote his book, A Troublesome Inheritance, where he declared the three principal human races, white people, Black people, and Asian people, and they not only evolved to be different physically, they also evolved to be different socially, and that explains why only white people support democracy and why there can't be any democracy in Africa, because African people are genetically prone to violence and chaos. And this was at one point a best-selling book, just like The Bell Curve. Even before that, for a decade, he was writing in the New York Times, the chief science journalist of the New York Times, all of these ideas. And so I was very shocked and alarmed when I began to read these, and curious about how it could be that we continued into the 21st century to have genetic scientists doing their research based on this completely discredited false view of human beings. And so I ended up writing a book, Fatal Invention: How Science, Politics, and Big Business Recreate Race in the 21st Century, in order to just document how race was continued to be used as a genetic category, and these new explanations for how to discover race in the human population structure, and how to look for genetic reasons for racial health inequities and other kinds of inequities in America and globally. and also the rise of very specific biological remedies for these inequities that were supposedly caused by innate genetic differences. So I think it's my book and also other scholarship I engaged in since writing Fatal Invention in 2011 that I was asked to serve on this committee. So here's just a list of some of the committee members, and I think it's important and interesting to see that they really are a diverse mix of backgrounds. Many genetic scientists, some chief genetic scientists like John Novembre and others. Sarah Tishkoff from here at Penn. The two of us have collaborated over the years at Penn, and it was nice to be on the committee with her. Bioethicists, like Sandra Lee and others, anthropologists like Lance Gravely, and biomedical researchers as well, like Katrina Armstrong, just to give, and sociologists like Anne Morning. So a very diverse group of social scientists, historians, biomedical researchers, and a nice also core of very respected genomic and genetic researchers as well. So the statement of task, which we were asked to follow, was to assess the use of race, ethnicity, and other population descriptors, including ancestry. And those were the ones we mainly focused on. Race was a big part of our work, but as well as ethnicity and ancestry, and also being careful to define the meanings of those different terms, and how they were being used in genetics and genomics research, to develop best practice approaches for the appropriate use of population descriptors. We were supposed to provide a guide for genetic and genomic researchers and others who use genetics in their research, like biomedical researchers, for example, to discuss the obstacles to this. One of the questions is why in 2023 did we have to still be preparing a report on this topic, and why hadn't other task forces been able to succeed in ending some of the bad practices in genomics and genetics research, and then suggestions for how to implement our recommendations, and as I said, potential implementation strategies that would enhance the adoption of them. I have to say, since this consensus report, I'm on a listserv that looks at race and genetics that we alert each other about studies. And it's very disappointing to see studies continue to treat race as if it were a genetic category being published, just in the last few weeks. But we hope that some of the strategies we suggest will over time help to implement the best practices that we're suggesting. And then there were certain tasks that were out of the scope of our what we were supposed to do. We weren't supposed to get into the use of race, ethnicity, and other population descriptors in biomedical research. That actually is the task of a committee that's convening right now. And if you're interested, you can go to the NASEM website and see videos of the hearings, or I don't know if hearings is the right word, the meetings that they've had with experts coming in to inform the committee about the work, and we're eagerly waiting for that consensus report to come out. And we weren't supposed to address racism in science and genomics. We took that to mean, the racist biases of researchers in science and genomics. Although, personally, what does it mean? I always say that racism created race, and you can't discuss the meaning of race or how it's used without addressing racism. But that was the task. My understanding is they meant not to do research into studies of racial bias in science. And then we weren't supposed to provide policy recommendations to NIH and government agencies. NIH was funding us, and you'll see that we did discuss the OMB categories and their implications for research. But I suppose you could say those recommendations were for researchers and how they should interpret the NIH policies more so than telling NIH what we thought NIH should do in terms of its policies. And so we defended why this report was so urgent right now, not only why we needed another task force considering these questions, but also why it was needed now. I'll just go over a few of them. One was just the urgency of addressing the confusion over the meaning of race and its use in genetic and genomic research. We were very aware that this continued to be a very heated controversy in genomics research and needed to be addressed. We thought that genetics investigators are not always clear on why they select certain population categories in their research. Why use race at all? Why not use ancestry? What do they mean when they replace race with ancestry? A lot of confusion over this, with some genomic researchers realizing that race might be controversial, simply replacing it with the word ancestry or ethnicity, or using race/ethnicity in order to get around it, but not really getting into the problems with using these categories. There was also, we knew, an effort to include more diverse groups of people, including different racialized populations, in other words, non-white people, in genetics and genomics research, to recruit people from diverse population groups into genetic and genomics research. Why? What's the purpose for that? And also that there were more advanced methods, continually advancing methods for analyzing genetic variation, and also the failure or limits of previous investigations and task force to actually make a lot of difference. I'm getting this from the nifty NASEM graphics that were created, and you can find more on the website. But one thing we wanted to point out was the very notion of population, what that means, and that it's always something that researchers make up. They decide what grouping of people. There's no natural populations. The researcher determines what to call a population and what its boundary can be and who belongs to that population. And it's not the same as a label, a specific group label used to describe a population. Those labels are not written in the genes. You have to decide what classifications you're going to use and how to determine what they are. And then we also noted that researchers often use these population descriptors inconsistently. And I'll say some more about that, what I mean by that. But sometimes mixed population descriptors in one study as if they're all the same type of grouping of people. As I'll point out, we ended up being very critical of the use of race as a proxy for human genetic variation. Okay, so that indicates some of the problems we were dealing with, but again, number one, you'll see this comes out of our number one recommendation. Race has been inappropriately used as a measure of human genetic variation, and it continues to be used that way despite all the evidence that it shouldn't be. And course, we present evidence of that in the report. That's a scientific problem because racial groupings don't map onto and cannot capture the complexities of human genetic variation. And it's also a social problem because that belief about race and its use in scientific studies has sent the message that socially constructed groups are actually objective, natural divisions of human beings. I think genetic researchers, as well as doctors, I think, are two of the main people that promote this view because the public believes that they know what they're talking about, that they are using objective scientific methods to just classify human beings into racial groupings. I think this is not what we were charged with because we didn't look into biomedical research and medical practice, but I'll just add that from my own experience, doctors have historically, from the time of the Enlightenment and the slavery era, reinforced this view and also are taken as people who are experts on how human bodies work. And so their continued use of race as if it were a natural division of human beings is very powerful. Genetic scientists and doctors, in my experience, are the ones, and they also tend to be the most resistant to changing their ways. Okay, but I'm adding that. You didn't say that before. Okay. We identified this kind of thinking as typological thinking. The idea that there are types of people that genomic researchers are discovering, as opposed to realizing that genomic researchers bring in certain assumptions about how the human populations are structured. And that kind of thinking has negative consequences, just both because it facilitates false views about race, but it also supports racial hierarchies. And it also, especially in genetics, leads to the view that differences that are caused, including health differences, by social inequities like structural racism and experiences of racism can't be changed because they're written in genes. And that also is a very harmful way of thinking about inequities. Okay, continuing with that, the failure to account for how population differences reflect differences in environmental exposure. All the genetic scientists on the committee emphasize that genes alone do not determine anything. Their genes are always interacting with the environment, and many studies left out the environmental exposures that are reflected and cause population differences, especially in health, but in other realms as well. As I mentioned before, this variable, inconsistent, and unreflective use of population descriptors is harmful to the science itself, because how do you determine the accuracy of a study? How do you replicate a study if people are using different population descriptors to reach conclusions that are supposed to be based on the same kind of evidence? So referring to race, ethnicity, and ancestry interchangeably, mixing different types of group labels in a single study. I'm sure many of you have seen this, where a study will be comparing some outcome between groups, and they're using ethnic labels as well as national labels, as well as religious labels, as well as geographic labels, all in one study. This also, I teach a course on race science and justice here, and we were just looking at ancestry testing, or discussing ancestry testing companies and how they present their results, and they often mix it up. Like they'll say you are 10% African American, and 10% Finnish, and 10% Jewish, and 10% white, and 10%, and whatever remaining percent East Asian. Like they'll mix up all what, and they use national labels all the time to tell you what your ancestry is. But what does it mean that your ancestry is British? You know, as I said to my class, my mother was from Jamaica. She considered herself British, but I don't think that's what these companies mean when they say my ancestry is African American. Okay. And so these are the main population descriptors we considered and we referred to in the report. And really importantly, we took time to define these different population descriptors, which often get, as I just pointed out, get mixed up with each other. Sometimes they're used interchangeably when they mean different things. And so you can see ancestry has to do with lineage and descent. Geography is a spatial location. Ethnicity often refers to cultural similarities. Indigeneity has to do with ties to a particular geographic location, as well as culture and traditions. And race, we emphasize, is a socio-politically constructed system that is that often ranks human beings and also is determined according to subjective beliefs and about shared, not actual shared ancestry, but subjective beliefs about shared ancestry based on perceived innate biological similarities. We pointed out that sometimes, and this is a problem with NIH requirements that require for some clinical researchers to categorize their research participants, that those categories, even stated by the Office of Management and Budget, which just 2 weeks ago updated the categories to include Middle Eastern, North African, and also to reemphasize that these are social categories, but often genetic researchers treat them as if they are natural biological categories. And we also took pains to distinguish genetic ancestry, genetic similarity, and race, and made it clear that genetic ancestry has to do with the actual lines of your ancestors, an individual's actual genetic heritage, not a predetermined group, social group that someone decides, either you or the researcher decides you belong to, based on these perceived made up ways of categorizing people. Ancestry is a real, a real line of inheritance of DNA. And genetic similarity, which we found very helpful as a way of thinking about what many genetic and genomic researchers are actually looking at. They're actually looking at the similarities of different individuals at the genetic level. And so we preferred that, and the genetic researchers had a lot of input on this. They really like this idea, both as a description of what genetic researchers are and genomic researchers are really looking at, but also as a way of moving away from race as the be all and end all of comparing human beings and assuming that somehow that comparison tells you something about people's genetic ancestry. Okay, so this is the structure of our report. In the first section, we delved into the past and current use of population descriptors. Looking at research about how genetic and genomic researchers were actually using these descriptors, but also we went quite a bit into the history, which I started with, of where these ideas came from in the first place. What does race mean? How was it invented? Why was it invented? What is the history of the use of the biological concept of race and all the horrific harms that it has justified over the centuries? And we also noted that there are multiple approaches to the use of population descriptors in today's genomic and genetic research. So then we got to our recommendations, and we decided to, and I'll say a little bit more about this in a minute, to have guiding principles, a very kind of bioethical approach. Sandra Lee was very influential on this, that we couldn't give too detailed recommendations because there's so many different ways in which genetic and genomic researchers use population descriptors. And so we decided to present guiding principles that would guide researchers in the future, but also that we gave a little more, and I'll show you a little more guidance than just the principles, suggestions of how these principles would apply to the use of population descriptors in different major types of genetic and genomic research. And then we ended with our recommendations for implementation and accountability in an effort to try to urge people who are stakeholders and have influence over this to take our recommendations into account. Not only the researchers, but others who influence what researchers do. Okay, so we have the principles: respect, beneficence, equity and justice, validity and reproducibility, transparency and replicability. Those were the guiding principles that we decided on, and go into depth about what those mean. And then we had certain requisites that we thought all researchers should apply to their research, which was avoiding typological thinking, measuring environmental factors, and engaging with communities and participants. So this was another important aspect of our deliberations. We took into account that not all communities have the same view of what ancestry means. We heard, for example, from people from Indian or Native Indigenous communities about the fact that ancestry and kinship may mean something very different and have different kinds of ways of determining it that rely on certain cultural traditions that most genetic and genomic researchers don't use and may even think are not appropriate. But we thought it was important that genomic and genetic researchers engage with communities that they're studying to determine what their meanings of connection, human connection are, and to take that into account. Now, we don't go into great detail about how you would do that in every single study, but the point is it's important to do that, to not believe that the scientist is the only one who is equipped and expert and qualified to determine the meanings of ancestry, especially, we focus especially on ancestry. But also the meaning of ethnicity and who belongs to that population that's been identified by the researcher. Then we, in our guidance, we divided up the kinds of research that genetic and genomic researchers do into 5 categories: gene discovery, trait prediction, cellular and physiological mechanisms, health disparities, and human evolutionary history. And I'll get in a moment to how we provided a rough guidance for researchers in these different fields, because we determined that these population descriptors would have a different kind of significance in each of these different ways of using genes and genomics in research. And then for implementation, we identified 5 groups of people that would be influential on making sure that our recommendations are hopefully encouraging. Our recommendations should be actually put into practice. The study participants, the funders, and I should say these are in addition to the researchers themselves. The point here is what can help researchers, support researchers in implementing our recommendations. So the study participants, funders, journals and professional societies, research institutions, and journalists and the media. I think this is something, well, I think all of these are, and the combination of them are, make our report different from most reports on this topic. I think some reports might borrow from one or the other, but to have guiding principles as well as to recognize there are different types of genomic and genetic research and the kinds of deficits that we highlight, as well as thinking very carefully. We have a big part of the report on what is it that researchers themselves need and what can push them toward implementing these ideas. These obviously, many people have been writing about this, for example, saying, do not use race as a proxy for human genetic variation research. People have been writing about that, for decades and decades, and it hasn't worked. Just writing about it hasn't worked. And why? Part of it is that there's so many influences on researchers. Where can they get their funding? The NIH requirements for the use of race in identifying research participants. A huge influence. I know when I was doing research for my book, Fatal Invention, I talked to a number of genetic scientists and biomedical researchers and asked, why did you use race in your study? Why did you classify people by, why did you report your findings by race? And many of them would either say, I'm not really sure what race means, or I don't think it's a proxy. So why did you do it? And the simple answer is, the NIH requires me to do it. That's why. So the huge influence. Journals and professionals, why do journals publish studies that are so flawed, that don't even define what race is? They don't even tell how they determine which race participants belong to, or they mix up different categories. They leap to conclusions. I often use a hypothesis in a study that stated Black race, independent of other factors, increases the risk of preterm birth. Well, I don't think that should have been published. What does Black race mean? And how did you determine what that was, independent of all other factors? What? That's impossible. But even if it was possible, at least have 100 factors, or just have 2 and then say, oh, it's because of Black race that Black women have higher rates of preterm birth. Okay. And I could go off the media. We know that the media influences researchers, and I've seen this as well. If you're going to get a headline in the New York Times, if you say studies for, in fact, that study got a headline. Study points to genetics or to genes in racial differences in preterm birth. If they didn't find it, it wouldn't have gotten a headline in the New York Times. So I could. Okay, let me keep going, because I could go deep into that. Some cases I know of where researchers changed the conclusion because they knew they'd get, a big headline in the paper. Okay. So the guiding principles, I I'm not going to go into the meaning of each one. I think hopefully, because that would be a whole lecture, but hopefully you can understand respect for the study participants, beneficence, thinking about the outcomes and of your particular use, equity and justice, validity and reproducibility, transparency, replicability. I've talked about all of these. And so we decided to use guiding principles because we knew that the field is dynamic, different, you'll come to different conclusions depending on the particulars of the study, the community you're working with, et cetera. All right. So then our recommendations, we had 13 falling into 3 categories. The requisites, and I talked about 3 of the main ones, the guidance, best practices, and then the implementation and accountability. And I want to emphasize 2 of the conclusions. So we reached conclusions, and then based on the conclusions, we had recommendations. And so our one of our first conclusions was that race is neither useful nor scientifically valid as a measure of the structure of human genetic variation. And just plain as day, researchers should not use race as a proxy for human genetic variation. I was very happy about that. Okay. So guidance for researchers. As I said, we identified these types of genomic studies and then had different kinds of guidance for each one. Noting that genes are used differently in these types of studies. And researchers have an obligation to think about carefully what the population descriptors they choose are, what they mean, and what are the implications of using them, and to be clear in the study how they're defining these categories that they're using and why. Okay, be transparent about it. That was the principle of transparency. Okay, so I'm not going to go into, I don't have time. I want to end shortly, but we felt that, okay, we were guiding principles, but we should put some meat on the bones of that. And so this is, I'll be honest, this is the work of the genetic researchers. We had, the others had some input in it, but these details about when different descriptors would be appropriate in different types of studies was really the hard work of the genomic and genetic researchers. And so one thing I'll point out is that race, so there were just a few times we said that certain categories or descriptors should not be used at all. And you'll see that for race, in almost every single category, we said it should not be used at all in the study. The only one that we said perhaps could be appropriate as proxies for environmental, but not genetic effects. So if you use race, it's a proxy. It's not a proxy for genes, though, because again, we said you can't use race as a proxy for genes, would only be in health disparities research using genomic data. But you'd have to make it clear that you're not using race as a proxy for genes. Okay. And so you can see that there, in many cases, we said, okay, you could use it, but you've got to really think about the descriptor. And you can also see from this that our favored descriptor was genetic similarity, as I mentioned before. Okay. So the requisites for sustained change, which I mentioned briefly before, avoiding typological thinking, measuring environmental factors, and engaging community participants, and all of these are mapped onto particular recommendations. And so you can see for engaging communities, for example, that researching should include experts in community engagement to integrate the perspectives of students throughout the research process. And then these important players in determining which research gets funded, which research gets popularized and published. We have recommendations for all of them, which map onto the guiding principles and the requisites that I mentioned before. Don't publish research that doesn't abide by the guiding principles, basically. Don't fund it. That's the shorthand, more complicated in the report. And so I think I've made all of these points. So I'm going to go to the end, which is for more and the very detailed description of the guiding principles, the list of recommendations, and the advice to the stakeholders and influencers is available at the NASEM site. And also, I don't know if you can click onto this, but you can go to the website. The staff has created a lot of helpful resources. I just want to point out one, which is an interactive resource that is designed to help researchers figure out whether they should proceed with a study if they've got a wrong-headed view of how to use these population descriptors, or proceed. But you see that some of them are don't proceed with the study if you haven't followed the steps that we recommend in the report. So I'm going to end there and left a little time for Q&A. And again, the report itself has lots of information. The website has lots of resources to help navigate what is pretty complicated, I think, when you get down to the brass tacks, except for, I think, that one recommendation about race. Not that it's going to be easy to change, but I think it's a pretty clear-cut recommendation. Yeah. Okay, so.
Okay, we are open to questions from anyone in the audience, including folks who are joining us online.
Yeah,
That was really interesting. Thank you so much.
sure.
I'm just wondering if you have any recommendations or any kind of insight into how people who might be like lower in the hierarchy of their career field might
Yeah.
be, able to take this information and apply it without, causing a risk for
Yeah,
themselves in their professional
right.
life.
Yeah, well, to me, that's one of the chief benefits of this report. You have a report that was funded by NIH. It's a consensus report of leading experts across a realm of disciplines, including genetics and genomics. And it is published by the National Academies of Science, Engineering, and Medicine, which many people feel is, one of the highest and most qualified associations to speak on an issue like this. So you can point this out to whoever is supervising you that I'm backed by this report. And they can hopefully also, as a result of the report, there will be changes in research, as well as people who do research on how to implement the report, who expand on what does it mean for publication, as well as what does it mean to actually implement, that complicated chart that the genomic researchers put together. So there were some question marks to be looked at, but as I said, this is going to take time for these changes to be implemented, but hopefully it will be easier for the rising generation of researchers to implement it. I say all the time, I don't think it's going to be easy for people who've been doing this research for, 30, 40 years to make changes. One problem we came across, and we discuss it in the report, and also it's part of this interactive resource, where's your data coming from? We recognize that a lot of the databases already divide the samples into race. Years ago, Dwayna Fulwiley, who is an anthropologist at Stanford, wrote an article called The Molecularization of Race, where she notes that the DNA coming into the lab that she was embedded in, writing her dissertation, came in already labeled by race. And so the researchers were already using racially labeled data. Now, what do you do about that if the data that's available to you is already grouped by race? Now, of course, the researchers there went on to call the data coming from Black people and white people opposite DNA. They didn't have to do that. Yeah. She also found that they would do change up the algorithms over and over again. Why are you doing that? Because we have to find differences or we won't be funded. That's what they told her. I love this article, Molecularization of Race by Dwayna Fulwiley. We won't be funded if we don't find differences. So we're going to keep going through these. And then they developed the PURE database, where they tried to have pure African DNA and pure Caucasian DNA. And I believe that lab is still doing that to this day. So how do you change that? It takes a while, but thank you for your question, because that's what it's going to take, is for the next generation that sees the flaws, the scientific as well as justice flaws in the way genetic and genomic research has been conducted. Not that all of it has been conducted that way, but much of it, and demands a change. And now you have something to say, no, I want to do it this way. And that's what this consensus report says we should do.
Thank you.
Sure. That's a great question. Yeah.
I know you specifically called out that the point of this wasn't to evaluate this in the context of biomedical research and clinical care.
Yeah.
But what do you think the trickle down effects or implications of this for those fields?
Yeah, I think it's really important. And in some ways, this next committee was seen as a follow-up to our committee. So I hope that some of the fundamental ideas in our report, for example, that race is a socio-politically invented grouping, will apply to their deliberations. However, I don't think that's necessarily true from my engagement with the committee. I spoke to the committee, and there is this sense that I got, again, this is subjective, that some people on the committee might believe that you have to take race into account in order to address health inequities, that there's certain differences between races that are manifested in different rates of disease that require attention to possible biological differences. They just seem more open, maybe paradoxically, I don't know, more open to the idea that there may be, in some cases, some biological difference that is causing health inequities that, it would be wrong not to take them into account. And so I'm not really sure. Plus, the day I spoke, there was also a genetic researcher whom we didn't invite to speak to our committee, who was, has a history of writing articles that, at least in the past, I mean, maybe he's changed, but he didn't, he seemed to support a view that maybe there are genetic differences between races that explain health inequities. And he's certainly written that in the past, and downplayed it a little bit in his, what he had to say, but it was still fundamentally there, I think. And I don't know if that's a sign of anything, but the chair, this is all public, so I'm not revealing anything, but the chair, at the end said, thank you so much. We need a diversity of opinion. We need a diversity of opinions, in this committee, which I suppose is true, except that should we have a diversity of opinions that contradicts what our report said from a genomic scientist? So again, all my interpretation, but I'm anxious to see how it turns out. I don't know. I spoke at the very beginning of their deliberations, and they've been meeting, and those I'm not privy to. So, but hopefully it'll have an impact. I think ours was very strong, especially on race and its distinction from ancestry. And I mean, that should be relevant to the biomedical researchers as well. Great question.
So we are unfortunately at time, but we are so appreciative of you coming and sharing the committee's work and your work that preceded it. Thank you.
Thanks so much.